I left the term organic chemicals up here because a lot of times when we hear the word organic, we think of organic food source. We associate that type of organic connotation in with this. Organic chemicals are a little bit different. It's not necessarily what you would assume an organic compound would be. For something to be considered an organic compound or an organic chemical, it has to have carbon. In it. And so let me give y'all some examples here. So, let's see, we've got water, oxygen gas, carbon dioxide, table salt, and then we'll do methane, CH4, methane. Based off of your definition of an organic compound, well, let's say not based off our definition, if you had not looked at that definition and I were to ask you about this list, what on here is organic? Most of us would say water is absolutely organic, right? Oxygen gas, the air that we breathe, organic. Carbon dioxide, organic. Table salt, we might have been a little iffy on that one. Methane, most of us would not associate methane with being organic, but look at your definition of organic compound. Contains compounds containing carbon bonded to hydrogens. Do you see carbon and hydrogen here? So this is not an organic compound. No carbon and hydrogen here, no carbon and hydrogen here, no carbon and hydrogen here. Methane is carbon bonded to hydrogen. So methane would be considered an organic compound. Glucose, which is one of our main sugars, C6H12O6. Doesn't have carbons and hydrogens in it. Yes, this would be an organic compound. Yes. Uh-oh. It's not working.
I asked them what they thought about being pioneers. To use a tablet.
Oh, I can help with that.
Okay, come fix that right.
I'll be right back, y'all.
Guys, all four of y'all.
Organic chemicals, right? Not necessarily what you would think. Carbons and hydrogens is what you've got to have. You can have other stuff in there too, but you got to have carbons and hydrogens as part of it. So none of these will be organic.
What was that formula? You didn't say it was formula of
This is a sugar. Glucose and fructose both have the same shorthand. 6 carbons, 12 hydrogens, 6 oxygens, C6H12O6.
Is that like their functional group?
Do what?
Is that like a functional group?
No, this isn't a functional group. This
Okay.
is
Just having trouble understanding
just like
that.
any of these. H2O is water, C6H12O6 is glucose, O2 is oxygen gas, carbon dioxide, table salt. So it's just a name for a different compound. It's one that we'll go over today.
Oh, wow.
We'll get to it in a moment. We're close. This is just a visual on how versatile carbon is. You can see all the different types of bonds that you can get with carbon. Carbons and all the associated hydrogens, carbon double bonded to various things, oxygen in this example, carbon double bonded to a nitrogen, carbon to carbon to carbon. This is diamonds, right? You have all carbons down together, that's what diamonds, or how you make diamonds. Carbon double bonded to carbon, triple bonded to nitrogen. You have carbon chains, so just one long, hundreds long chain of carbons with all the associated hydrogens. You can have carbon chains that have branches on them, and we can even get ring structures. So carbon is extremely versatile, and we find it in a lot of the compounds that are associated with life.
Is that something we have to know? I mean,
You just need to know that carbon is versatile.
okay, good. Thank you.
Now, you've got a table in your book that talks about functional groups. And the functional groups are something that you will have to know. So if you're at that table, mark down the functional groups. I want y'all to be able to recognize them. And so be able to associate a name with the functional group. And then the second bullet point down here that says can be found in, be able to associate the hydroxyl group with alcohols, associate the hydroxyl group with carbohydrates. So know what types of molecules that you would find in each functional group. Now, what a functional group actually is. If you look at your carbohydrates, your lipids, your proteins, and your nucleic acids, they all four have the same basic structure. They're all carbon-based, like a carbon-hydrogen skeleton. And then what you add to that carbon skeleton determines if that carbon skeleton is going to be a protein, or if that carbon skeleton is going to be a lipid, or if it's going to be a carbohydrate. So functional groups, they do what the name implies. They give the carbon skeleton a function. If you add an oxygen down to a hydrogen, which is called a hydroxyl group, if you add that onto a carbon skeleton, it makes that carbon skeleton, that compound, it turns it into an alcohol. We also see that the hydroxyl group is one of the functional groups that we find on carbohydrates. So there's other functional groups that are part of the carbohydrate. But the hydroxyl group itself will turn something into an alcohol. Depends on, we got a bunch of different alcohols. So if it's a very small carbon skeleton with a hydroxyl group, that's one type of alcohol. If it's a huge carbon skeleton or a ring structure with a hydroxyl group, that's a different kind of alcohol. So you've got a lot of variation with that, yes.
I know there's a lot of different functional groups. Are we going to need to know like specific ones?
What do you mean?
Like what you just pointed out for OH, it's going to make it into an alcohol.
Yes, all of them.
Okay.
All the functional groups, you'll have to be able to do that. Associate, need to be able, if I give you a structure, which
Okay.
I will do here in a moment, and ask you what functional group do you see, you'll have to be able to look at that structure and say, okay, well, I see a hydroxyl group, and then you mark hydroxyl group.
Okay.
Or if I asked you, to match up your functional groups with the macromolecules or the molecules that they're found with, almost like a matching section, could you link those up and say that the hydroxyl group would be linked with alcohols? The hydroxyl group might be linked with carbohydrates. There's a couple of carbohydrates, but be able to associate that, which will make more sense when we actually look at the structure. It's not hard, right? It's just some thought into it. Being able to link this to this to this. So your next one is the carboxyl group. If you have a carbon skeleton and you see this mess right here attached to it, this is called a carboxyl group. This is a carbon double bound to an oxygen with a hydroxyl group attached. So carbon double bound to an oxygen with a hydroxyl group attached. This whole thing together is called a carboxyl group. You'll see this with your lipids mainly. The proteins, it says proteins, organic acids. We do not go that in depth to where you're going to be seeing these, the protein organic acids. So right here, if you've got your notes, circle the fatty acids. That's the association I want you to be able to make, is that the carboxyl group is the functional group that you're going to be able to see in the fatty acids on your lipids. And again, I'm going to show you all pictures of that here in a moment so you can put a visual with it. Your amine group is probably the easiest to pick out. So these are what you're going to associate with proteins and nucleic acids. The amine group is the only one that has nitrogen in it. And it is. You find it as part of the structure for your protein and part of the structure for your nucleic acids.
Can you go back one?
Oh, sure.
Yeah, I did.
Okay, alright. Next functional group is the ester group. So carbon double bound to an oxygen with another oxygen attached, and then what's after that doesn't matter, and what's before that doesn't matter. It's some type of carbon skeleton. But this right here, the R stands for remainder. So it could be anything attached here, is what the R means. But this is the important part. The carbon double bound to an oxygen with a single oxygen off the other side. This one is also lipids. When we look at our glycerol head and our lipids, you're going to see this structure there. The sulfhydryl group, this one is the only one that has sulfur in it, so it's also an easy one to pick out. I do not have y'all learn the 20 amino acids. I don't have y'all do the structure for those. But the amino acid cysteine has sulfur in it, has the sulfhydryl group in it. So amino acids are associated with proteins, so specifically cysteine, but your broad group here is going to be proteins, is the part you need to be able to associate proteins specifically. Now, both of these are carbonyl groups. One is a terminal end, one's an internal end. That kind of implies where it's located, terminal versus internal. The carbonyl group specifically is a carbon double bound to an oxygen. That is the carbonyl group, the carbon double bound to an oxygen. If it's at the end of something and there's nothing to the, so right here, this is probably your carbon skeleton, and then you've got the carbon double bound to an oxygen, and there's a hydrogen attached because you got to fill the empty spot, but then there's nothing else past that. So it's at the end of the molecule. It's on an end of a bonding site. That's what the terminal end means. Internal means that you've got your carbon skeleton, you've got your carbonyl group, and then you've got some other stuff that happens after that. But your carbonyl group is still there. It's just in the middle of a set of bonding sites. Both internal and terminal carbonyls show up in the polysaccharides. So that would be starch, glycogen. We'll do a little bit with cellulose in here. Your peptidoglycan layer. There's a couple of others I'm missing. Chitin. The terminal end is only found in the aldehydes. You do not need to worry about that. If you're Xing stuff off your list, X that one off. You don't have to be able to associate that with an aldehyde. And you do not have to associate the internal end with a ketone.
So we just no go problem with the polysaccharide.
With the polysaccharide, yes. With the polysaccharide. You X off aldehyde, X off ketone. And your phosphate group, right? This is an important one we see a lot this semester. The phosphate group is also easy to pick out. It's got a central phosphate and then several oxygens and hydrogens surrounding it, but the phosphate is in the center. This one has a charge to it. This one has a negative charge to it. Your phosphate group has a negative charge. This one becomes important because where we see the phosphate group is in the sugar phosphate backbone of DNA and RNA. There's one place, there's a couple others. The sugar phosphate backbone in DNA and RNA. Because the phosphate group in DNA and RNA has a negative charge, it gives DNA or RNA a negative charge as well, which gives the cell an overall negative charge. Well, we work a lot with cells in the lab, and a lot of our techniques that we use in the lab to identify microbes have to do with staining techniques or some type of microscopic examination where you're putting a cell on a slide and looking at it. And the reason we're able to do all of those things is because the cell has that negative charge. Your stains that we use are designed to have a positive charge so that they're attracted to that negative cell. The microscope slides are designed to have a positive charge so that negatively charged cells will stick to the slide and not wash off or rinse off or anything like that. ATP, which I've looked at in A&P and biology, is an adenosine and 3 phosphate groups. So 3 negative charges, and that's how those negative charges, those negatively charged phosphates are what give ATP all its potential energy. And we'll look at ATP today, I hope. But negative charge, don't forget, negative charge on the phosphate. Okay, let's see here. A lot of this I've said as we've gone through. Table 2.4 is an overall list of all of the macromolecules. Your carbohydrates, all the different ones that we'll be going over, the lipids, the different ones, same with the proteins and the nucleic acids. Here is where I'm going to go ahead and have y'all either put stars or mark off some of these things that we will or will not cover. And then we're going to go over them. Your carbs, which are your sugars. There's different kinds, right? There's single sugars, which saccharide. What does saccharide mean?
A single sugar.
Yes, saccharide is sugar. And then mono is 1. So your monosaccharides are 1 single sugar. We will go over the monosaccharides, so you can put a big star by that one. The disaccharides, we will do 2 of these. Sucrose and lactose. Sucrose and lactose. Your polysaccharides, which are your large, multiple sugar carbohydrates, are actually a big one for this class because the cell wall of bacteria is made from, a good chunk of it is made out of polysaccharides. And when we look at how to target the microbe, that's one of the things that we focus on because that's something that we don't have. We don't have a whole lot of polysaccharides in our cell wall, our cell membrane. And so that's a difference that we can target. So we do spend some time on the polysaccharides. So you'll have everything on this list we'll go over. The lipids, yes on the triglycerides, yes on the phospholipids, yes on the waxes, no on the steroids. So no on the steroids. Proteins, the polypeptides are a yes. Nucleic acids, all 3 of those are a yes. DNA is a yes, RNA is a yes, ATP is a yes.
Can you say those again?
The nucleic acids?
Well, the whole table.
Everything on this list is a yes.
Okay.
Proteins, the polypeptides, yes. Nucleic acids, DNA, RNA, and ATP are all yes. We'll do all of those. Okay. Alright, let's walk through our carbohydrates. We'll start with those. So, carbohydrates are our sugars. Look at the word carbohydrate. What can you pick out in this word? What element is carbo probably referring to?
Carbon.
Carbon. And then what about hydrates? What does that make you think of?
Hydrogen.
Water. Water is right. Hydrates, water. CH2O. Carbohydrates are all multiples of CH2O. A carbon, a set amount of carbon and oxygen, so the same amounts, with twice as many hydrogens. So always multiples of CH2O. So one of the things I'm interested in y'all being able to do is identify if what you're looking at is a carb or a protein or a lipid. And how you know if what you're looking at is a carb. Even if you don't know what carb it is, if it's in this multiple, then it's a carbohydrate. So if I ask you, say if I give you C55, H110, O55, and I say, what type of macromolecule is this? You don't know what this is. I just made this up. I don't even know what this is. But what do I know?
It's got carbon and the hydrogen.
That it's a carbohydrate, right? It's got twice as many hydrogens as it does carbon or oxygen. It's in multiple of CH2O. So it is a carbohydrate. I could give y'all C80, H160, O80. Yeah. And you won't know what this is, but you will know it's a carbohydrate because it's in the multiple of CH2O. So they're all in that multiple. Now, our monosaccharide probably the big one. So saccharide, sugar, mono means one. These are your single sugars. Your main monosaccharide, and they are showing it here without labeling it, is going to be glucose. Glucose is one of the only ones that you'll know the chemical shorthand of because it's just so widely used. You probably already had this one before in AP or biology, I know. But glucose is C6H12O6. That's how many carbons, hydrogens, and oxygen that it has. Glucose is a monosaccharide, and it's the sugar that every organism follows metabolically as the most ancient pathway. So everything is able to metabolize glucose. Not everything can metabolize certain polysaccharides. Not everything can metabolize a protein. And not everything can metabolize a lipid. Everything is able to process glucose in some way. It seems to be the most ancient pathway that we have. When we study metabolism, we're going to study it off of glucose and then talk about where the other macromolecules come in. And microbes, we feed them a lot of glucose because we know that they all can process it and they all like it and they're all going to grow from it. It has a symbol. A lot of times this is too much for people who write textbooks to constantly write, so they use symbols. The symbol that they use, and they're showing it to you there, probably not quite as accurate. I can draw that very well. That's your symbol for glucose. So when you see that, that means glucose.
Well, I can't draw it out.
I know. I feel like my drawing skills need some improvement. You have some other ones that we'll see this semester: fructose, the lactose, sugar, and mainly those are going to be associated with our nutrition section for our microbes. What you're going to find is that as we learn how to identify our microbes, we can identify them based on what types of sugars they like to ferment with. You have, so like your milk, for example, there is a lactobacillus, the bacteria that we find in milk. And that bacteria is the one that's responsible for making your milk taste funny when it's gotten too much age on it. If you leave the lid off, or after enough period of time anyways, and it gets access to oxygen, it will start to ferment the lactose in the milk, and the byproduct that it produces is lactic acid, which we associate with our muscle being sore, and it's the same one. But when you drink it, it tastes funny, and we don't like it. And so, depending on what microbe you have, if you can, we have different agars that will turn colors depending on if lactose is being fermented, or if it's citrate being fermented, or what sugar it is. But if you know what sugar they like to ferment, then it helps you identify them because they all do something different. So glucose is the main one. Fructose and lactose are two others. There's fructose. Fructose looks very similar. And then lactose is down here. I do not want y'all to spend time trying to learn these structures. You will not be asked anything about these structures. But if I ask you to identify a monosaccharide, if I give you a list of glucose or fructose or ribose, if I give you, and then I throw starch or glycogen in there and say, which of these is a monosaccharide, could you pick out off of that list of the names which one is a monosaccharide? So that's what I'm looking for y'all to be able to do, is to be able to identify if this is a monosaccharide or a disaccharide or a polysaccharide. I will not ask y'all about structures like this. Okay? Maltose is one I told y'all we didn't need to look at, but sucrose and lactose I did want to spend some time on. Not necessarily on the structures, but the sugars that are used to make up these two disaccharides. Glucose and fructose are how you get table sugar, which is sucrose. So,
Can you repeat that again?
yes, glucose and fructose are how you get sucrose, which is our table sugar. Sucrose is equal parts, right? 50% glucose, 50% fructose, so it's one of each, and they come together in a bond, and that's how we get our table sugar, our sucrose. So one of the things that just comes up in our daily life is high fructose corn syrup, right? That's been pulled out of a lot of foods. It was a pretty big topic as far as health goes. In the past few decades, we've shifted away from it to now we're focusing just on sugar in general, sucrose in general. But high fructose corn syrup, the reason they say that it's high fructose is where table sugar, sucrose, is a 50-50 split. High fructose corn syrup is 45% glucose, 55% fructose. And that's why they call it high fructose, because it is higher than what table sugar has. Glucose, our bodies process, and microbes as well, process it, like I said, very well, and process it as a sugar and store it as quick energy. Fructose is a little bit different. Some microbes can process it, some can't. As far as we go, as a human, fructose for us gets processed as a fat, not as a sugar. Something about it, our bodies don't process it as a sugar. It stores it as a fat. Lactose, the sugar in milk. We identify some of our microbes using lactose because some of them can ferment and some can't. And we also use the lactase operon as an example when we do our genetic engineering lab. Some people are lactose intolerant. Microbes are the same way. Some microbes are lactose intolerant. They can't ferment lactose, can't metabolize it, and then some aren't, and so we use that as an example when we get to that one. How you get lactose is a galactose and a glucose. Put those two together, and that's what gives us lactose. So again, don't focus on the structures, names, right? We're looking just for name association. Let's see here. Where are our polysaccharides? Well, are we missing? Do y'all have a slide that I don't have?
I'm looking at it.
Do you have a polysaccharide slide that I don't have? I don't really have anything on polysaccharides here. Polysaccharides. Let's see. So this is cellulose, which is one of the polysaccharides. Poly means many, saccharide sugar. So what polysaccharides are is they're where you have multiple single sugars all linked up in a big long chain. we move into actually look at our microbes specifically in chapter 4 and 5, we will pull specific polysaccharides out and take a look at them. Especially when we were doing our chapter 4 on bacteria, when we get into their cell wall, our antibiotics that we give people target the cell wall of microbes. That's their first, when they come up to a microbe, the antibiotic comes up to a microbe, that's its first line of defense. It's got to get through the cell wall to get in and do its job. And so a lot of the antibiotics have something in their weaponry to be able to target that cell wall. And a lot of times that's enough. If you poke holes all in the cell wall of a bacteria cell, well then it loses its integrity and that's it. But sometimes you need a little more than that, so that's just the first step. But the cell wall is made up in part with polysaccharides, which are a bunch of single sugars linked up. Cellulose specifically is one that we'll take a look at when we get into that chapter. And cellulose is a big target because that's something that our cells do not have. We don't have cellulose, we don't have a cell wall. We do have a cell membrane. Bacteria have that as well, but that's not something we can target since we have that as well. We gotta focus on the differences. When we get into those chapters, we'll pull some more of these out specifically. But what a polysaccharide is it's where you have a whole bunch of individual sugars linked up. So not just 2, not just 3, many, right? Many sugars linked up together in various ways. There can be branching, there can be big cyclic ring ones. This right here with cellulose, these are chains of glucose molecules that hydrogen bond with each other and form a wall. So this chain will hydrogen bond with this chain, will hydrogen bond with this chain, and make a wall. And then our antibiotics have to get through that wall, right? So for now, just understand that that's what a polysaccharide is. Okay, lipids. Lipids are going to be discussed also with our microbes because when you start looking at the lipids that are embedded in the cell wall of some of the microbes, they're what give us all of our like toxic shock syndrome symptoms. We're very allergic to some of the stuff that they have in their cell wall, which are mainly lipid-based things. And so we'll give some specific names once we get to that point in that chapter. For now, let's just get the basic structure of a triglyceride down. This should be something y'all had in A&P, something y'all had in biology. Triglycerides, which are lipids, right, fats, have a basic structure that includes a glycerol head, and 3 fatty acid tails. And that's what you're seeing on this slide here. So they turned it sideways, but you've got in red or whatever color that is your glycerol head with 3 fatty acid tails. And here they are all attached. Do y'all see where you're having your, where you're getting the carboxyl group here? The carbon double bonded to an oxygen with what was a hydroxyl group, but you had to dehydrate the hydrogen off to bond. But do y'all see that functional group present there? Absolutely, we see that, don't we? Yes. Do y'all remember what the difference is between saturated and non-saturated on your lipids? Anybody remember that? So, these fatty acid tails are carbon skeletons. Right? It's a carbon with hydrogens bound to it. Carbons and hydrogens bound to it. All 3 tails are. When you have a saturated fat, so a saturated fat is one that's going to be solid at room temperature. When you have a saturated fat or a lipid that's a solid, what that means is that all 3 of the tails have as many hydrogens as they're supposed to have. They're fully saturated with hydrogens, meaning that every carbon has 2 hydrogens. And the last carbon has 3 hydrogens. That's the max they can have. If you have a tail one of your tails that has a carbon to carbon double bond at some point in there. Where they've only got one hydrogen instead of two. If you have one of the tails that has a double bond in it, between one of the carbons, what that does is it unsaturates that tail. This tail is no longer as saturated with hydrogens as it's supposed to be. And these carbons, it's not that they're missing one, but yeah, they're missing a hydrogen because they bonded twice with each other. That eliminated the ability to have another bond here with another hydrogen. And so this would be an unsaturated tail, which would make this whole lipid an unsaturated fat. So instead of being a solid at room temperature, now it would be a liquid. Well, how does this double bond change that? Well, when all of the tails have single bonds between the carbons, all of the tails go straight down. When there's a double bond here like this, I didn't draw it right, but when there's a double bond here like this, it makes the tail kink out this direction at that double bond. When the tail kinks out, and the microbe, or even in our bodies, is trying to store this lipid, if the tail's kinked out, the lipids will not store as close together as they're supposed to. They won't pack solidly together and be a solid. There will be gaps between this lipid and the next because the tail's sticking out. And that's what makes it a liquid instead of a solid at room temperature. And they're showing you that here too. The other lipid, so this was the triglyceride, right? Very easy to identify. The glycerol head, 3 fatty acid tails. Tri for 3 fatty acid tails. Glyceride for the glycerol head. The second one is the phospholipid. The phospholipid is also something you'll have before, right? It's in the cell membrane. You're going to see it in the cell membrane with your microbes as well, and it looks very similar, but a tad bit different. You still have the glycerol head, and you still have fatty acid tails. But instead of 3 fatty acid tails, you only have 2 tails that hang below the head. The other difference so this is your triglyceride. With a phospholipid, the third tail is actually still there. But with a phospholipid, the third tail has a phosphate group inserted at the top of the third tail. Phosphate groups have a charge, right? Negative. That phosphate group makes the tail, instead of go straight down, it makes it wrap around the glycerol head. That phosphate group makes the third tail wrap around the glycerol head. Well, lipids are supposed to be hydrophobic. Now, what did we say that meant, right? It's not necessarily water-fearing like we're taught. What does it actually mean to be hydrophobic? It can't dissolve. Can't dissolve in water, right? Water's not going to be able to dissolve it. It also, why can't water dissolve a lipid? Why is a lipid hydrophobic? What is missing from the lipid for it to be able to be dissolved in water? Charge, right? You have to be charged for water to be able to come in there and pluck you apart, right? Water is based off of charges. And so lipids don't have charges on them. That's what makes them hydrophobic, because water has nothing to attract to pull the lipid apart. Does the phosphate group have a charge?
Yes.
Yes, we have just added a charge to our lipid. So the phosphate group, this tail that wrapped around the glycerol head has a charge to it. So as this tail wrapped around, what it did is it made the head of this phospholipid hydrophilic. So a hydrophilic head and the tails are still hydrophobic. So the phospholipid actually has both properties. It's got a hydrophilic head and hydrophobic tail. That's amazing that the phospholipid does that. Because what that means for us is that the phospholipid behaves a certain way. Whether it's our cell membrane or a microbe cell membrane, the phospholipid is what makes up the cell membrane. Y'all remember that? A phospholipid bilayer is what makes up the cell membrane. These phospholipids, because they have a hydrophilic head and a hydrophobic tail, will behave a certain way in water. You can take a jar full of water and put a handful of phospholipids in it and shake the jar up, and every time the phospholipids will come back to the same orientation. And we used to have one of these jars. One of us dropped it and broke it. We don't have it now. Every time, the phospholipids will put their heads on the outside and put their tails in the center. So the heads have a charge and are attracted to the water, so they all orient outward towards the water. The tails basically repel water because they don't have a charge, and so they all orient towards the center. The way it's usually explained is that the heads are protecting the tails from the water because they're hydrophobic and they're water fearing, right? It's not really what's happening, but that is what it looks like. And so you'll shake it up and they go all about and they come back to the same orientation. Now in your cells, in microbe cells as well, they do the same thing. They'll line up with their heads outward and their tails to the inside in a double layer, right? That phospholipid bilayer. And so if you continue this around, this would be your cell membrane, and this would be the inside of the cell. And it's a double layer. And there's a control mechanism here, especially with our microbes. It's super important because their environment constantly changes, right? They're kind of at the mercy of their environment. The temperature change, the pH change, whether there's food or not, salt concentration. So they've got to have a control here on what goes in and out. And that phospholipid job is to control that. And it does that with its charges. Things with a charge, molecules with a charge, are not going to want to cross this uncharged area that these tails create. They're going to need some assistance. Whereas things that don't have a charge are going to be more compatible with crossing this barrier because they're going to be unaffected by that. So we'll get to all that later as well, but that phospholipid will be really important. And that's the difference in what it looks like. So do you think you could pick out the triglyceride versus the phospholipid?
Yes.
The difference between the two? Perfect. And that's steroids, that's not good. Okay, proteins. The last two sections are proteins and nucleic acids. Now, proteins, how you get a protein is from your DNA. That's what your DNA is coding for. So I like to cover the nucleic acid section first. So let's skip ahead to the section on nucleic acids and we'll come back to proteins. All right, what do y'all remember about DNA and RNA? Let's see what we remember. Let's start with something simple.
DNA.
What does DNA stand for?
Nucleic acid.
Yes. I asked the biology class that this morning, and they're like, I had higher hopes for y'all. Yes. Deoxyribonucleic acid. DNA. Deoxyribonucleic acid. What does RNA stand for? Ribonucleic acid. remember about the differences? And most of it's written on this slide, but what do y'all remember about the differences between DNA and RNA? What's the big difference? Thymine and uracil is a big difference, right? DNA, if you look at the nitrogenous basis for DNA, DNA has adenine, thymine, guanine, and cytosine. Adenine and thymine like to bond together, right? A pairs with T, and G and C like to pair up. RNA has 4 nitrogenous bases, has adenine, but does not contain thymine, has uracil instead, still has guanine and cytosine, but your base pairs are a little bit different there. We're going to practice this here in a minute. What else is different? A big difference. How many strands do they each have? DNA is double-stranded, right? DNA is double-stranded. Whereas RNA is single-stranded. Now, we're taught this. We're taught this in many classes, and then you're going to come in here, and we're going to get to our virus chapter, and what you're going to find about viruses, which according to our rules are not actually living things, but have DNA, have RNA, and do some other things. They don't metabolize or reproduce on their own, but they do reproduce using a host cell. But what you're going to find is that everything we're fixing to talk about DNA and RNA does not apply to viruses. They have DNA and RNA, but they have single-stranded DNA and double-stranded RNA and positive and negative sense DNA and positive and negative sense RNA. All the rules just go out the window. Viruses just have their own little world going on. But for everything else, these rules seem to be applying. Right? Let me give y'all, see how we remember. I'm going to raise this up for just a second. We'll just write it right here on the side. Let's see. Okay, let's say that this is one side of a double-stranded helix. So this is DNA code, this is one side. If I asked you, could you give me the complementary side? Following your pairing rules, could you give me the complementary side? What would pair with A here?
T,
T,
A, C, G, C, T, A, T.
Everybody comfortable with that part? So, what DNA actually is, you've got your 2 sets of code here. DNA is a code, right? It's not just all these letters that don't mean anything. Your DNA, microbes DNA too, all the DNA, it's a code. Every 3 letters means something. It's a triplet code. Every 3 letters codes for an amino acid. And amino acids, there's 20 different ones. There's 20 different amino acids, and those amino acids are what make up proteins. So what is actually coded for in all genetic material, whether you're a virus, a bacteria, a fungi, a worm, a dog, a cat, an elephant, a person, a fish, every living thing that we know of, including the non-living viruses, follows the same code. So, the problem with DNA is that cells don't speak DNA. Microbial cells, human cells, animal cells, does not speak, it's foreign language, does not understand DNA code. So, when the DNA needs to be read, the first thing that an organism has to do is to transcribe that DNA strand into an RNA strand. The cells speak RNA, not DNA. They understand that language. So, do you think you could do that? Do you think you could transcribe this bottom DNA strand into an RNA strand? What would that look like? start with the T. What would I put here?
U,
Nope.
Okay.
A,
You put an A, right? What pairs with T?
A.
An A. So this is a common mistake I see people make, is we assume, because of the way it's written, it's very misleading. We assume that you replace thymine with uracil. And that's not what that says. It says that uracil will bind with adenine, not thymine binding with adenine. A still binds with ribose because RNA have A in it. Yes, so A will still pair with T. But what's going to pair with A? Not thymine, right? Because RNA doesn't have thymine. RNA has, what does RNA have? Uracil. So here you would put a U. And then G with C, C with G, G, what would I put with thymine? Right here.
What? The T?
What would I put with thymine is not a U.
And an
A.
An A, right? A is what binds with thymine. What would I put with an A?
A U,
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 U.
and then A.
A.
Got
Y'all
it.
see that?
Mm-hmm.
Are you sure you got that?
RNA.
Yes, this would be an RNA strand.
Makes sense.
Now, the cell can read this. It speaks RNA, and it can go in here, and it can read every 3 letters and associate those 3 letters with an amino acid. And
So I have a
so,
question. So you just basically just binding like the on the RNA, you just basically binding what they combine, what it binding with, like the A, which is, for example, the T, and since you don't have the T, it automatically bound with the A.
yeah, you pair it with its complement. So, what do we know that's attracted to thymine? Adenine, right? What is attracted to adenine? Uracil. What's attracted to cytosine? Guanine. What's attracted to guanine? Cytosine. That's how, when the cell reads this, how it identifies what to pair with the T is because it looks for what's attracted to thymine. Whatever's attracted is going to show up here. Whatever's attracted to adenine is going to show up here. If RNA had thymine, thymine would be here, but RNA doesn't have thymine. What it has that's attracted to adenine is uracil.
I get it.
So it can still read it, it just can't write it. That's how I think of it. Anyways.
Sure. Sure. Now, let me show y'all. This is one of the places where I digress. But this is some of the stuff I did my graduate research on, and I just find it so fascinating. And I think y'all do too. We have figured out what the key for the code is. So every 3 letters means something. And it's RNA code, right, not DNA code. And so this is the table that shows the key for the code. On the code that I just had written on the board for y'all, the very first code that was listed was an AUG. And so you look on here and find AUG, and it tells you that in an organism, the triplet code AUG in RNA codes for the amino acid methionine. CCC, that triplet code codes for the amino acid proline. CAU would be histidine. Arginine is any of these. My point with this code is that how amazing is that, right? This is what your genetic code is actually coding for in every living thing that we know of, even in viruses. And maybe that's an important point to make, is every living thing that we know of at this point and with our knowledge reads this same code. If you take CCC in a bacteria, a virus, a fungi, a worm, a dog, a cat, a horse, antelope, you name it, an insect, CCC will always code for proline in every organism. So this code is universal. That's good and bad, right? Good part and bad to it. I guess the bad part would be that's how microbes make us sick. So viruses, for example, viruses, when they infect you, they infect your cells. And what they do is they take their DNA, their RNA, and they inject it into your cells. Well, because everything reads the same code, your cells don't know that that's viral code. It just knows that it's code. Hey, I need to read this and I need to do what it says. It's probably important. And so your cells read it and build whatever it says to be built in the code, which the code says build more viruses. And so viruses turn your cells into viral factories that build toxins and different enzymes that are harmful to us, and building more viruses to go out and infect more cells. And it's able to do that because the code is universal. Everything reads the same code.
So how do you stop that? I mean, I know what you're saying, it's universal and it reads it in our body. I guess that's what medicine comes in.
Well, and your body has some, we get the common cold every year, which is a virus. And are you sick forever with it? No, your body eventually gets a handle on it. And so your body's got some built-in mechanisms where it recognizes that this is a foreign something in our body and we need to target that. And you have an immune system that's its job.
I'm sorry. Yeah. I have another question. Yeah.
Some viruses like HIV, what makes them so hard for us to get under control is that they hide in our bodies and they hide from our immune system. They're very smart viruses, and so we have to use medicine to help. We have to use other things to help with that. But yes, medications, they work the same way that they do as with bacteria. You got to find something that's different about them to target. And when we get to viruses, you'll see that because they have weird differences with their genetic material, it's something we can target. For example, the HIV virus is a reverse transcriptase virus. And so what's supposed to happen is you have a DNA strand that gets transcribed into an RNA strand and then translated into a protein. And that protein does something, good or bad, it does something. HIV has an RNA strand in it. It doesn't have any DNA. That virus doesn't. And so it injects an RNA strand into our cells, plus an enzyme to read the RNA strand. That enzyme reads the RNA strand, turns it back into a DNA strand, and then hands us the DNA strand and says, okay, now read it. And that's something that we can target because our cells don't do that. Our cells go in the right order. And this reverse transcription, this reverse transcriptase enzyme, if we could find a way to target it, that's where a lot of the research is at. Because that's one of the big differences. And viruses, they all got some weird thing like that where there's a difference about them. Yes?
Well, I have a question. What about people with including myself as well, that have autoimmune disease?
Mm-hmm.
Where the body just attacks its own cells. So how does that play in with that?
Well, I have a friend who also has an autoimmune disease. He has lupus. And some of it, depending on what type of autoimmune disease you have, some of it they've figured out a few clues on what causes it and why the body's suddenly turning on itself. You have some research that suggests that a virus is behind it in some way. Others suggest that maybe the prion, we've talked briefly about the prion, right? It causes mad cow and Creutzfeldt-Jacobs. You have
I have rheumatoid arthritis.
Okay. Most of them, we don't know. We don't know yet what causes a lot of the stuff that we have. A lot of the mental disorders, a lot of the autoimmune disorders, we don't know yet. We haven't figured it out. If there is some microbe or non-microbe causing it. Something has caused your body to not behave like it should. Whether that's genetic or microbial related. They don't know. Now, that's the bad side of the code being universal. We also see that antibiotic resistance shows up that way as well. And so, because everything reads the same code, we can do things in the lab like genetically modify an E. coli cell to have a jellyfish gene in it that makes it glow in the dark, right? We can do weird stuff like that because the code is that universal. But microbes use that as well, and if they come across a code on another organism that gives that organism some cool ability, like resistance to an antibiotic or the ability to produce a certain toxin or something like that, they will swap genetic code. They do that. It's called conjugation. And it's scary to see it, honestly, because they're swapping their genetic information. And so you'll have a microbe who was susceptible to an antibiotic now be resistant. And y'all probably seen this yourselves. You've got maybe even 2 kids who are sick with the same thing, and this antibiotic worked on this kid, but it didn't work on this kid. And that's because there's some extra something that particular strain is picked up, or it mutated in some way. And a lot of it has to do with the fact that this code is universal.
So now they're saying that the new studies that with the DNA, you can determine what child birth you want, whether it's a girl or boy.
Yeah, we talk about that later on.
Is that true?
That's not a new thing, but yes, one of the things that it's not legal here in the U.S. As far as I'm aware, it hasn't been made legal yet, but it is legal in the UK where you screen embryos, and it's usually not abused. It's for people who they have genetic disorders of some kind, and they want to try to have a child without that child having that genetic disorder. And so they take the woman's eggs, the husband's sperm, they put them together out in a lab, put the egg and sperm together and do it for like multiple embryos. And then once the embryos have formed and are just a ball of cells, they screen them. And they say, okay, well, this one's going to be a girl, boy, this one's going to have the genetic, this one's going to have these genetic mutations, this is going to have genetic mutations. And they give you a profile on each one. Part of what they give in the profile is the gender, if you choose. And so you get, basically, you choose which embryo you want to implant, which child you want to have. And the purpose behind it is good intended, because it's trying to prevent a child from being sick that's born. But you have a lot of people who think that that's even worse than abortion, because you're not just discarding one embryo, you're discarding all the ones that you didn't pick. And it's not legal to do that here. But even now, when you get pregnant, you can go in and they take a DNA sample and they can immediately tell you if it's a boy or a girl before the baby grows big enough to tell that on an ultrasound. And they can do that with the eggs very early on. But it has to be done in the lab, right? It can't be after you're pregnant. It's a different test. Did that answer your question? But yes, there's a whole like ethical side to all that we'll talk about when we get in that cloning section on what's allowed, what's not, and just kind of scientifically, we are way ahead of where we are ethically, I guess I should say. We have the technology to do a lot of things that are not being done because we as a society cannot decide how it should be regulated. And until they're able to decide, they've just kind of said, we're not going to do it until we can all agree on how it should be handled. I don't think we'll ever agree on it, though.
What about people with I know.
No, that's why we're here. Go on.
But what about the kids that are born with autism? You know, what's up with their DNA sample like that?
There's different causes for autism. Some they've identified, some that they haven't. You know, there was a group who thought the vaccines, and we talked about that, I think. But most of what has been identified is the genetic link, where there's some mutation. There's one, for example, called Dravet syndrome, that they know exactly. They can, if a child starts having seizures and they're suspecting autism or something like that, they have a reason to suspect, to check the child, they go into their genetic code. And I heard of the Human Genome Project. Heard of that? So there's a group of researchers who went in and they took several different ethnicities, backgrounds, and they took DNA samples from everybody, and they went in and mapped every single letter in our code and figured out what the correct code for the person should be. And so now, if you're sick and they can't figure out why, they take a sample of your DNA and they compare it to what the correct code should be, and they can see where your mutation is. And they say, okay, well, now we see that you have a mutation on P53, protein 153 or whatever, which its function is here in the liver, and that's what's making you sick because that protein is not functioning right. And so they have a code to compare to.
So it all looks like this code here.
This code right here. And so they can go check children with autism that way too. There's certain forms of autism where they've identified it's on gene 6, on chromosome 22, that they're 3 base pairs in, there's a mutation that happens right here that makes this child have this. And they go check, and sure enough, that child has that mutation. And then the next child will come in with the same symptoms, and they go check, and they say, well, this must be a different mutation. We have more we have to learn. And so they're slowly working through and identifying where all the errors are. And then, so the next question that usually comes up is, well, now that we know where the error is, it's nothing to go in and fix it. To take out the wrong letter, put in the right letter. Should we do that or should we not?
Is it ethical?
No, that's the question. Isn't that the question? And so you have some who say, yes, I would want that for my child, and you have others who say, no, my child is a gift and I wouldn't change anything about them. And so, I don't know. I don't know what the answer is. And so, they don't do it. They don't change it.
Okay, I'm clear.
No, I found that really interesting. So yeah, that was so very interesting.
Now, the good parts about the code being the same is that's how we get a lot of our pharmaceutical drugs. So insulin, for example, we've gone into our DNA, our RNA, and we found the section of our code that codes for insulin production. We made a copy of it, pulled it out, and we presented it to E. coli cells. And since everything reads the same code, those E. coli cells take up that code for insulin. They say, okay, got some code I need to read, let's do what it says, which says make more insulin. And so you go to these pharmaceutical companies, they have these warehouses with these vats in them that have all these genetically modified E. coli cells that are producing insulin, and then we harvest that. And that's how we get a lot of our drugs. And it's very clean, very efficient, and very cheap for the most part. Not that we know it for what we pay.
I was gonna
for
say, if it's so cheap, why is insulin so expensive?
Because they know you have to buy it,
And that's why people go to Canada.
Well, hopefully with the new law that'll change.
Do what?
The new thing that was passed to reduce costs of medicines here in the U.S. to be not more than,
Oh, you try to think that Trump just passed through some law in that okay.
yeah, it's supposed to reduce the costs.
He taking out the middleman, that's what he said.
Yeah.
all right, now, one thing I didn't touch on, and this is not anything that y'all have to know right now. We're gonna do this later on in like chapter 10 or something. But, pull this back up. Can y'all see through it enough to see the code? No? If I read, perfect. If I read AUG, and then from a cell, and I read these 3 letters, and then these 3 letters, and then these 3 letters, I would get a particular set of amino acids in a certain order. But what if the cell started reading here? Would that be a complete different set of amino acids? So how is the cell supposed to know where to start reading?
Is it like keeping their bond? Is it like, because it's not binding, right?
Say that again.
You know how you said that the A and U bind together? So it's not like they're binding. Go ahead.
So how does the cell know where to start reading? Methionine is, they put it in green because it's the start code. Every code starts with AUG. That's the correct reading. So what a cell will do is it'll read the RNA string until it finds AUG. And then it knows, hey, I need to start a triplet reading right here. And then read every 3 letters after that. And it will keep reading until it gets to one of the stop codes. And then it knows, hey, this is the end of that protein. Cut it off, we're done here.
And there's 3 of those.
There are. And again, this is not stuff you'll have to know right now, but it's very interesting and it helps us understand what a protein is.
You say right now, so I'm gonna learn it now.
Yeah, you'll learn it later.
G and G A.
But we'll do this in chapter, I think it's chapter 10. We'll practice with this time.
And you had just answered my question when you said that.
Oh, good. Okay, I've got like 5 minutes here on proteins and we'll be done with that section. We're good with nucleic acids, right? DNA, RNA, we're good with all that. I don't have y'all on any of those structures, don't worry about that. We'll do that later on. ATP will do later on. Okay, back up here to protein. So, the code that we just looked at codes for 20 different amino acids. So far, we have not identified more than 20. If you Google it, there may say that there's 21 or 22, and that's because there's a couple that we've synthetically created in laboratory settings. Naturally, there's 20 different amino acids. And so the combination that you get for a strain, right, the amino acids are what we're coding for in the genetic material, is endless. The combinations are endless because you take those 20, and there's no limit to how many. You can have a strand of amino acids that's 1,000 amino acids long. Not only that, but the different combinations of order that you can have those 1,000 amino acids in is endless. And so you see proteins, very small proteins, and then we see large proteins like hemoglobin. Hemoglobin is, if you take the weight, it's a 1 million atomic mass units inside. It's huge. And it's made up of amino acids. And so one thing I do want y'all, I don't want y'all to know the individual amino acids. Not interested in that. What I do want y'all to have, don't do a good job showing. Is this, we'll just use this picture. The basic structure that all amino acids have. That's what I'm interested in. So this right here, this colored box, ignore it for now. This part up here in blue, and the black part here, this part up here is the basic structure that all amino acids have. Every amino acid has a central carbon with a hydrogen attached, has an amine group over here, so a nitrogen and 2 hydrogens, and a carboxyl group over here. So a central carbon with an amine group and a carboxyl group attached. Every amino acid has that basic structure. Now, what determines the 20 different ones is what you attach to that structure. So this basic structure, if you attach a carbon and 3 hydrogens to it, that's going to give you alanine. If you attach this mess to that basic structure, this gives you valine. So what you attach to the basic structure of the amino acid gives you the 20 different ones. I don't need y'all to know the 20 different ones. I want you to know the basic structure. The whole point of your DNA is to sequence the amino acids so that they can be put together in a chain in a particular order. So that means that amino acids have to be bound to each other, right? The first one's got to be bound to the second one, which is bound to the third one, which is bound to the fourth one, and so on. The bond between amino acids has a name. It's called a peptide bond. A peptide bond. And you've got to dehydrate the hydroxyl and the hydrogen off, so a dehydration reaction. And then that puts the 2 amino acids together. So the carbonyl group and the amine group are what bond. So you don't see 2 amine groups bonding together. And you won't see 2 carbonyl groups bonding together. The amino acids have to be turned in a way that a carbonyl and an amine group are beside each other. And then the peptide bond forms between those 2. If you like the written part, stick on these 2 slides, but I'm going to look at some pictures here. proteins run organisms, whether it's people or microbes. What runs our bodies is proteins. And because that's what's coded for in your DNA, right? It's amino acids which form proteins. What makes one protein different from the other is the sequence of the amino acids. Well, what that really means is the shape of the protein. Y'all heard the term shape determines function, right? That's always referenced with proteins. Proteins are made up of the same 20 amino acids. Their shape is what determines how they're going to function and what job they do. And so one of the things that we go over is how protein gets its shape. There's 4 different levels to protein structure. And they're titled, thankfully, in a way that makes sense. The first level is called primary structure. The second level is called secondary structure. The third is called tertiary, and the fourth is called quaternary. So they make sense. Let's walk through what happens. Let's look at the first one. Oh. I hate it when they switch to new editions. I always lose pictures. Okay, so this up here is primary structure. What primary structure is? It's the very first thing that starts to determine the shape of the protein. Primary structure is the sequence of the amino acids. So what order are they in? That's the very first piece of the puzzle that starts to determine the shape that the protein will eventually take. The sequence of the amino acids. That's primary structure. Secondary structure. Secondary structure, depending on which amino acid is beside which amino acid, they're going to interact in certain ways. Some of them interact in a way that forms alpha helices, which you're seeing here. It looks like a helix, makes the strand wind up like that. Some amino acids, when they get together, they make the strand do this orientation. It's called a beta-pleated sheath. So what secondary structure is it's the formation of alpha helices and beta-pleated sheaths. It's not that the strand will just form one or the other. You may, if you've got a whole strand of amino acids out here, these over here may interact in a way that forms a helix, and then 2 more down it's a sheet, and then a helix, and then a couple of sheets, and you're going to see some of both all along the strand. It just depends on who's next to who. But secondary structure is the formation of alpha helices and beta-pleated sheets. The third layer, or the third level of protein structure is tertiary structure. Tertiary structure is the first level where we start to see a 3-dimensional shape. And so in tertiary structure, the alpha helices and the beta-pleated sheets start interacting with each other, and they fold up. And how they wad up into a ball, the shape that that takes depends on how many alpha helices and beta-pleated sheets there are, and who's next to who. And so tertiary structure is the interaction between the alpha helices and the beta-pleated sheets. And this is the first level where we start to see that 3-dimensional shape. Now, I'm guilty of this because it's just the way we talk, but I misuse the word protein a lot because it's just a common generic term that we throw around. But there is a difference in a protein and a polypeptide. At tertiary structure, when you get that 3-dimensional shape at tertiary structure, that is a polypeptide. Right there, that's a polypeptide. Some polypeptides will be polypeptides and they're ready to do their job as a polypeptide, they're the right shape and they're ready to function. Some polypeptides go on to quaternary structure. And what quaternary structure is where you have several polypeptides that come together and form one large protein. So quaternary is optional. It's where several polypeptides come together and form one large functioning protein. So, do y'all have a mental flow of events going from DNA to RNA to an amino acid sequence, and then how that amino acid sequence folds up to give you a polypeptide or a protein? Can you visually walk yourself through that imagery? Yes, right? Yes. Y'all have any questions?
The quaternary structure is where several polypeptides come together and form a structure.
Form a large functioning protein. So this is a protein. By definition, this is a protein. This is a polypeptide.
Okay.
Okay, y'all don't have any questions? We got through chapter 2. And y'all can take a break and then we'll start with lab stuff. So 2:40.